Now entering into meiosis 1, we begin with prophase 1. This is where the homologous chromosomes will begin to condense. The chromosomes then begin to pair up based on length, gene by gene. Now you can see that the chromosomes have become physically attached to one another in a state called synapsis. Now that synapsis has occurred, crossing over will occur, where genetic rearrangement between non-sister chromatids happens. The non-sister chromatids have exchanged specific segments of DNA with one another. After this crossing over has occurred, you see a chiasmus being formed. A chiasmus is here where cohesion holds the two sister chromatids together. Now, as you can see, the centrosomes have moved away to become on opposite sides of the cell. Spindle fibers will begin to form and the nuclear envelope breaks down, which is in a process known as prometaphase. Now, for the last step here, centrosomes on opposite sides attach to a kinetochore, which is a protein structure at the centromere of the homologs. This happens at the very end of prophase 1, where the homologous pairs begin to move towards the metaphase plate. Now in metaphase 1, you can see the pairs of homologous chromosomes are arranged at the metaphase plate, having one chromosome facing each pole. Here you can see the end of anaphase 1. The proteins responsible for sister chromatid cohesion that we observed in prophase 1 have now been broken down. This allows for the chromosomes to separate. The chromatids then move to opposite poles guided by the spindle fibers. Now finally, at telophase 1, the spindle fibers disappear from anaphase and the chromosomes move to opposite poles remaining in their sister chromatids. Now both halves of the cells have complete haploid set of chromosomes. Cytokinesis will occur simultaneously with telophase 1. This forms two haploid daughter cells as seen in the visual. In an animal cell like this one, a cleavage furrow forms.
Meiosis 2 begins with prophase 2. Between meiosis 1 and meiosis 2, there is no chromosome duplication, so spindle fiber formation is the first step. Similar processes of prophase 1 occur, such as nuclear disappearing, chiasma formation, and spindle fibers moving chromosomes to the metaphase plate. In metaphase 2, the chromosomes are formed along the metaphase plate due to crossing over in meiosis 1. The two sister chromosomes are not genetically identical. So now in metaphase 2, the spindle fibers are attached to the kinetochores of each sister chromatid. Then in anaphase 2, the breakdown of proteins for cohesion occurs as in anaphase 1. Spindle fibers will then pull each chromatid to each side. Now in telophase 2, the nuclei form and chromosomes decondenses and cytokinesis occurs. This division will produce 4 daughter cells that each have a haploid set.